Well, thank you very much, Michael, my good old friend, and thank you also to the organizers of this wonderful meeting, and thank you also to Ryan for giving us a personal idea of what it's like to have Barth syndrome. Barth syndrome was first identified just over 30 years ago when Peter Barth reported an X-linked mitochondrial disease that was manifested by cardiomyopathy and skeletal myopathy and all of these other phenotypes that we now know are associated with this disorder. But the molecular mechanism underlying Barth syndrome was not known. And the first clue that enabled a molecular understanding of Barth syndrome was in the cloning of the gene in 1996 by the group of Toniolo and co-workers. And they identified this gene on the X chromosome, and they called it Tafazzin. Now, a really landmark study in 2000 by the late Peter Freken showed that the Tafazzin deficiency caused a defect in cardiolipin remodeling. And as Dr. Kagan has just shown, this resulted in a decrease in cardiolipin, an increase in the monolysocardiolipin, and a decrease in unsaturated fatty acyl cardiolipin species. Which of these biochemical characteristics of cardiolipin causes the defect, again, is not known. So here, we see what happens in remodeling. The cardiolipin that is synthesized de novo is deacylated to form the monolysocardiolipin, and then reacylated to form the remodeled cardiolipin, which now has more unsaturated fatty acids, and Tafazzin is the transacylase that carries out this step. And Michael Schlama then showed that there's a virtual absence of the primary species, L4 tetralinoleic acid species of cardiolipin in Barth cells. So there's essentially no L4 cardiolipin. Again, raising the question, is the lack of remodeled cardiolipin the cause of the disorder? So what I would like to address today is what is really, which cardiolipin defect is really the cause of the disorder? Is it the lack of unsaturated fatty acids? Or is it the decrease in cardiolipin, in total cardiolipin? Or the increase in monolysocardiolipin? Which of these causes the defect? So I hope to make 3 points in my talk today. What cardiolipin remodeling defect causes the pathology in Tafazzin deficient cells? And here we use the yeast model to answer this question. And because the answer to this is that it's not the lack of remodeled cardiolipin, the second question is, what then is the function of remodeling? Why go through this trouble if you don't really need the product? And then finally, because to really understand the basis of the defect in human cells, we would like to identify the cardiolipin remodeling enzyme, the phospholipase, in human cells. This is not a trivial question, and can we use yeast to address this question? So, because clearly, if we can block the deacylation step, we can then see what is the difference between cells that have the remodeled cardiolipin and cells that don't. Now, this is sorry that you can't see the reference that this comes from, but this is basically from a paper from Ed Dennis's group showing all of the PLA2's superfamily, and you're supposed to be able to see, can you see the difference between red and black? I'm not sure. But anyway, all of these phospholipases that are in red are present in humans. So there's a lot of potential cardiolipin phospholipases, tens of phospholipases. In contrast, yeast cells have only a single cardiolipin phospholipase, and that's the CLD1, cardiolipin deacylase. And again, sorry, I have a reference to a paper by from Sepp Kohlwein's lab, Baranek, that's down here, but you don't see it. Sepp Kohlwein is the one that identified this gene and shows that it's the single deacylase for cardiolipin in yeast cells. So the approach that we're taking is to develop hypotheses using the yeast model and then test the outcomes of these hypotheses in human cells. And we can do this because we know that the functions of cardiolipin and Tafazzin are conserved from yeast to humans. We showed a number of years ago that the yeast Tafazzin mutant can be, the defects in the yeast Tafazzin mutant can be rescued by expressing the human Tafazzin gene. And also that many functions of cardiolipin that were first shown in yeast are found also in human cells. And as I mentioned, there is just a single phospholipase in yeast that deacylates cardiolipin, and there are also a lot of, all of the genes for cardiolipin synthesis and mutants thereof have been very well characterized in yeast. And so this is the whole pathway. This is the pathway for de novo synthesis of cardiolipin, and these genes have been identified and knockout mutants are available, and here are the remodeling genes. So to address the question, what is the cause of the pathology in Tafazzin deficient cells? Is it the perturbation of cardiolipin monolysocardiolipin ratio or the decrease in unsaturated cardiolipin? 3 students in my lab collaborated to answer this question. This is Yiran Li, this is Tsuinqi Ye, his name is down here, and this is Wenjia Lu. They all have posters, so I urge you to visit their posters. So the cardiolipin, the CLD1 mutant can distinguish between these two possibilities because the CLD1 mutant has a decrease in the remodeled or unsaturated cardiolipin, but the CL-MLCL ratio is pretty much normal, in contrast to the Tafazzin mutant, where both are defective. So, what happens when we introduce the CLD1 mutation into Tafazzin mutant cells? And that's the experiment shown here. So this is an experiment where we plate serial dilutions of yeast cells. So there's 10-fold less cells going left to right. The top 2 panels are the controls. So in glucose, everything grows because the yeast cells are growing by fermentation. But in ethanol, they have to respire, and so we see differences in growth. I guess the lighting is pretty high, so you may not be able to see these differences. But basically, in the cardiolipin mutant CRD1, that's the mutant block here that has no cardiolipin, we can see almost no growth on ethanol compared to wild-type cells. Is it possible to turn the lights down in the front? It's okay. If not, it's okay. The CLD1 mutant, the deacylase mutant, grows pretty much as normal. So it grows out to 2 dilutions. The Tafazzin mutant has a growth defect similar to the mutant that totally lacks cardiolipin. And what happens with the CLD1 TAS1? In other words, we introduce the Tafazzin mutation, the CLD1 mutation into the Tafazzin mutant, and we can see rescue of growth. It'll be a little bit easier to see here when we look at chronological lifespan. So here we're looking at the number of cells as a function of incubation time in the various strains. So you can see that the Tafazzin mutant has a shortened lifespan compared to wild-type cells shown here, and also compared to the CLD1 mutant, the deacylase mutant, which is also similar to wild-type. So the Tafazzin mutant has a decreased chronological lifespan, but when we knock out the CLD1 phospholipase, we can see rescue of lifespan. So the deletion of the lipase rescues the defects of the Tafazzin mutant in spite of the fact that it has decreased unsaturated cardiolipin. So we can say that is not the defect in Barth cells, in Tafazzin deficient cells. So we published this outcome in JBC in December, and I want to acknowledge also that Stephen Claypool's laboratory came to the same conclusion in a paper that they actually published before ours. Okay, so what cardiolipin remodeling defect causes the pathology in Tafazzin deficient cells? We can say that it's the decreased CL-MLCL ratio and not a decrease in remodeled cardiolipin. So then what is the function of cardiolipin remodeling? Why do it? And the clue to understanding what is the function comes from looking at the regulation of the synthesis of cardiolipin and the regulation of expression of the cardiolipin biosynthetic enzymes. So here we looked at expression of each of these genes, and we looked in the different stages of growth, from early log to mid-log to early stationary to late stationary. And what you can see is the CLD1 gene is clearly the most highly regulated gene in the whole cardiolipin pathway. And expression is highest in the stationary phase. So what is happening in the stationary phase is cells that are growing in log phase in glucose are fermenting and getting most of the ATP from fermentation. And then as glucose is depleted and the cells have to respire, then the ATP has to come from respiration. And this is when you see the increase in CLD1. And concomitant with this increase, we see an increase in unsaturated CL species in stationary phase. So here is a mass spec analysis. This was done by Riekelt Houtkoper and Fred Vaz. And if we just focus on these two groups, these two clusters, we can see here, for example, is the more saturated species, which is decreased in stationary, and here's the more unsaturated species, which is increased in stationary. And similar, this very saturated species is present in log phase and almost non-existent in stationary phase. So CLD1 is increased in response to the need for respiration, and this occurs concomitant with the increase in remodeled cardiolipin. And further evidence for the role of respiration is seen during glucose starvation, where again the cells have to generate ATP from respiration. And you can see, again, the greater increase in CLD1 compared to all of the other genes in response to glucose starvation. Now, if CLD1 responded to an increase in respiration, then we would expect that cells that cannot respire would not exhibit this increase. And so that experiment is shown here. Here we're comparing CLD1 expression in Rho plus cells, these are respiratory competent cells, to Rho 0 cells, these are cells lacking mitochondrial DNA that cannot do respiration, so respiratory deficient cells. And we can see that the CLD1 increase in stationary that we see in wild type cells is not apparent in the Rho 0 cells. Now, if CLD1 responds to respiration, you might expect that the transcriptional activators that regulate expression in response to respiration may affect this gene. And looking at the promoter region of CLD1, we see that there are consensus sequences for two transcriptional activators that respond to respiration, HAP2 and MIG1. And so we looked, so if these activators regulate CLD1 expression, we would see altered expression in mutants of MIG and HAP, and that's what we see. So here we look at CLD1 expression in these HAP mutants that do not have the HAP activator, and we see clearly reduced CLD1 expression. And similarly, if we look at CLD1 expression in the MIG1 mutant, we also see about a 50% decrease in CLD1 expression. So we can say that both HAP and MIG1 regulate transcription of CLD1, again consistent with a response to respiration. So to summarize so far, increased CLD1 is observed during stationary phase. This corresponds with increased unsaturated cardiolipin. Increased CLD1 is also a response to glucose starvation when cells have to respire. There is no increase in CLD1 in respiratory deficient cells, and CLD1 expression is regulated by the transcriptional activators that respond to respiration. So what are the consequences of altered CLD1 expression? I mean, CLD1 is increased in response to respiration, but if you don't have CLD1, the cells can still respire. So what is it needed for? And in fact, expression of CLD1 is sometimes deleterious. It's deleterious if Tafazzin is not present, because deletion of CLD1 rescues the Tafazzin deficient cells. So this suggests that too much CLD1 is not a good thing, and we then look to see what happens when you overexpress CLD1. So here we look at cell growth as a function of time. And in wild type, this is the growth curve of wild type cells just expressing empty vector. This is the normal condition. And here are wild type cells overexpressing CLD1. Clearly there is a growth defect. There's also a respiration defect if you overexpress CLD1. So if we look at O2 consumption in essentially all the respiratory conditions, in wild type cells overexpressing empty vector versus overexpressing CLD1, you can see that overexpressing CLD1 causes a decrease in O2 consumption. Now, we expected to see that there would therefore be a decrease in ATP levels, but actually we saw the opposite. So looking at ATP, this is the cells overexpressing CLD1, there's actually an increase in ATP levels. How can that be? We reasoned that would happen if glycolysis was somehow upregulated to compensate for the decrease in respiration. And so that's actually what we see. So if we look at ethanol synthesis, which is the end product of fermentation, glycolysis and fermentation, in wild-type cells overexpressing empty vector versus overexpressing CLD1, we see an increase in ethanol in the overexpressing cells. So this leads to our hypothesis that cardiolipin remodeling actually controls energy metabolism by modulating respiration and glycolysis. So in response to respiration, we see an increase in CLD1 and then therefore an increase in remodeling. And this is followed by a downregulation of respiration and increase in fermentation. The corollary hypothesis, and this is really the hand-waving based on no data part of the hypothesis, that when there is an increase in respiration, well, we know when there's an increase in respiration, there's an increase in damaged cardiolipin. So we suggest, and this is where CLD1 is increased. So what we suggest is the function of CLD1 is to get rid of the damaged cardiolipin. And then the rest of the cycle proceeds. Okay, so the function of cardiolipin remodeling, we believe it's a response to increased respiration, and it triggers a shift from respiration to glycolysis, we hypothesize, and we further speculate that it functions to remove damaged cardiolipin. So now, to be able to investigate these characteristics in human cells, we need to identify the human phospholipases. And again, this is not a trivial question, but there are reports in the literature characterizing specific phospholipases. For example, IPLA2 gamma, there have been papers discussing IPLA2 gamma and the role in cardiolipin deacylation. So here are two papers from Richard Gross's lab, and in this, looking at the effect of ablation, genetic ablation of IPLA2 gamma in wild-type and Tafazzin-deficient mice. And if we look in wild-type mice, they showed a 15% decrease in cardiolipin and a 33% decrease in the remodeled cardiolipin, in the L4 remodeled cardiolipin. But it was not a total elimination of remodeled cardiolipin. And then also mitochondrial dysfunction. In the Tafazzin knockdown mice, they also demonstrated altered cardiolipin in monolyso and dilyso species, but did not prevent, the ablation of IPLA2 did not prevent the decreased cardiolipin in the mutant, that is seen in the mutant. Now, there are experiments with IPLA2 beta, and this is a paper that Mindong Ren and Michael Schlama published, and coworkers, and they looked at inactivation of IPLA2 beta in flies, in wild-type and Tafazzin-deficient flies. In wild-type flies, there was no effect on cardiolipin composition. In Tafazzin-deficient flies, they saw a partial reduction of the aberrant MLCL-CL ratio, and they show that ablation suppressed the male sterility defect, which is characteristic of Tafazzin-deficient flies. And to determine whether inhibiting phospholipase, IPLA2 phospholipases, would affect the Tafazzin cardiolipin phenotype, they treated Barth lymphoblasts with bromoenolactone and observed a reduction in the abnormal MLCL-CL ratio. So this suggests that clearly the IPLA2s are involved, and maybe this could be a target for Barth therapy. Another paper that I'd like to cite is from Ed Dennis's lab, in which they looked at enzymes that exhibit CL phospholipase activity in vitro. And these 4 phospholipases were reported. That's these shown here. And so the question that we would like to ask is, can we use our knowledge of yeast to definitively identify the cardiolipin phospholipases in human cells? And we initially focused on the IPLA2 beta and IPLA2 gamma because these are the ones that have been described in the literature. So here we see the Tafazzin mutant cells that are overexpressing either empty vector or the deacylase, the CLD1 yeast deacylase, or IPLA2 gamma or IPLA2 beta, the human genes. And what you can see here is, so this is the Tafazzin mutant just overexpressing empty vector, and these spots are I hope you can see them, are Tafazzin mutant colonies. If you overexpress the yeast phospholipase, CLD1, you see no growth of the Tafazzin mutant. If you overexpress IPLA2 gamma, you get a similar result to overexpressing the yeast CLD1, essentially no yeast colonies. And if you overexpress IPLA2 beta, you see almost no effect on plates. Thanks, Matt. This is maybe a little bit more clear in looking at the growth curve, growth of cells. So this top line is Tafazzin's mutant cells overexpressing empty vector. This is the control. If cells are overexpressing the green, the IPLA2 beta, there's only a very mild defect. And cells overexpressing CLD1 or IPLA2 gamma show a defect. Okay, so I'll just skip over this. So, can we use yeast to identify cardiolipin remodeling enzymes? We have shown that increased expression of IPLA2 gamma, similar to overexpressing CLD1, inhibits growth. And we think that we can use this as a bioassay to identify the human phospholipases. So, in progress are the following experiments. What is the function of CL remodeling? We're using what we in the business like to call the awesome power of yeast genetics to identify synthetic lethal interactions with CLD1. That is to say, we cross the CLD1 mutant to all of the 4,800 deletion mutants by a technique called SGA, synthetic genetic analysis, and thereby we can identify the condition which, under which CLD1 mutant is deleterious. We would also like to determine the mechanism of the shift from respiration to glycolysis and characterize the enzymatic activity of CLD1 with respect to damage versus wild-type cardiolipin. And can we use yeast to identify the human phospholipases? We think we can use this bioassay that I just showed you to identify the potential phospholipases and determine if inhibiting these enzymes will rescue Tafazzin deficient phenotypes. So with that, I would like to acknowledge the people who did this work. In my laboratory, Cunqi Ye, Wenjia Lu, Yiran Li, and actually Shuyang Chen did the initial experiment many years ago that we then pulled out of the drawer and seemed to fit in this story. And at Wayne State University, Mike Hutteman and Iksu Li, and our colleagues in Amsterdam who did the mass spec analysis, Ileana Chatzis-Pirou, Rikel Tautkoper, and Fred Vaz. And I'd like to acknowledge support from NIH, and I'm especially grateful to the Barth Syndrome Foundation and Barth Syndrome Foundation of Canada and Association Barth France for continued support. Thank you very much.
Thank you, Miriam, for this very informative and also very methodical talk, as always. We have time for questions. Yeah, sorry.
Hi, Dr. Greenberg. I was just wondering, why exactly did you choose to do the bioassay at 39 degrees for yeast?
Oh, good question. I should let my students answer the. Where are you guys?
So we tried different conditions to see whether their growth phenotype is different. Because in mild condition like 30 degree, there's less pressure on the cells, and a little difference in the cells may not show the difference. And if we use higher temperature, and there's more pressure on the cells, so if there's something defects in cells, it shows up more obviously.
Take into account of other defects that might be introduced with prolonged, incubation at 39. Like obviously, I see that there is, there's colonies versus no colonies. So that's pretty clear. But I guess, how do you account for like other problems? Like, do you need to get to 39 to see that dramatic of a?
This is a really excellent question. Let me just say that I only showed you the good data. I didn't show you all the plates where the data is not clear and you can't really see. So the cardiolipin mutant pretty much and the Tafazzin mutants grow pretty much at optimal temperature. They grow pretty well. So you have to stress them in some way to really make the phenotype apparent. And this is also very strain specific. So there are lots of physiological modifiers that affect the phenotype. So I think it's an important question. We don't know all of the physiological modifiers that cause this phenotype.
So thank you, Miriam. This was fantastic. So now I have a kind of a dual question to you. So you suspect that this yeast phospholipase is mostly utilizing as substrate potentially oxidized or oxidizable species of cardiolipin, which means that there should be enough of them to first get somehow oxidized.
That was your suggestion, actually.
Wow. Okay, I will take responsibility if you will answer.
Okay.
So now, but the second one also, this CLD, will it recognize oxidized species of cardiolipin? Because for calcium-independent PLA gamma, this has been demonstrated, that it doesn't need calcium because the way that it will recognize the substrate is by the substrate having this extra oxygen functionality.
So that's of course the $64 million question, which we don't know the answer to, and we hope you'll help us figure it out. Mindong.
So I have a question about the CLD1 overexpression. You showed that when you overexpress and you have some defect in growth. And do you have any data about the cardiolipin profile under these conditions? Because you would predict that they would have a less cardiolipin and more less cardiolipin in that case.
We don't yet have all of that data. I should ask you guys, what's the latest data that we do have on that? The CL and MLCL in the overexpression strain?
Yeah, we're still working on the experiment to test if that will affect CL to MLCL ratio. But the data we have on the cardiolipin profiles, but we did one time cooperate with Brad and Google, but we did not see that overexpression in CL will alter the saturation in cardiolipin, the fatty acid saturation in cardiolipin. But we are doing that experiment again to confirm the result.
Yeah, in progress.
Okay, one more question. Last one.
Hi. So the ethanol growth defect you see in the TAS1 mutant,
Yes.
is that for sure a respiration defect, or could that be an ethanol toxicity?
Oh, it's a very good question. It's probably a respiration defect because it's not only on ethanol. I mean, you see this on glycerol media and other respiratory carbon sources. So it's probably not an ethanol toxicity.